Much like how a car regulates its different parts, varying processes within the body are needed in order to keep it up and running for days on end. Like a car's mechanisms, these processes would require a means to do everything it does. As we explore and continue moving throughout the wonders of microbial physiology, we must all be familiar with a friend called metabolism. Long story short, metabolism is the main factor that composes all biochemical reactions that are occurring within the cell or organism. This study of bacterial metabolism would focus on the chemical diversity that is apparent in substrate oxidations and dissimilation reactions, all of which are necessary in the ultimate goal of energy production. In this video, we will discuss these main points: an explanation of respiration, fermentation, and the electron transport system, examples of microorganisms performing these different cellular processes, and discuss or cite applications of cellular respiration and fermentation to the society.
But before all of that, what is respiration? Well, respiration, or cellular respiration, is the breakdown of glucose molecules to release energy in the form of ATP, the energy-carrying molecule that powers biochemical processes. Below is the complete balanced chemical formula for cellular respiration. Note the single glucose molecule and 6 oxygen molecules lead to the production of 6 CO2 molecules, 6 water molecules, and energy in the form of ATP. With that out of the way, let's break it down. Cellular respiration has a collection of 3 metabolic pathways: glycolysis, the citric acid cycle, and the electron transport chain. So what is glycolysis? Literally, it means glucose splitting. Glycolysis occurs in the cytoplasm, where enzymes split a glucose molecule into 2 pyruvate molecules. Additionally, since it can be produced without oxygen, it is the only source of ATP for cells that can't undergo aerobic respiration. The process of glycolysis has 2 halves. The first half is the energy-requiring half, where glucose is split and uses 2 ATP, while the second half is the energy-releasing phase, where 4 molecules of ATP and 2 NADH molecules are released. Therefore, glycolysis has a net gain of 2 ATP and 2 NADH. The pyruvate molecules produced during glycolysis are transported to the mitochondria to be used in the second pathway. The second pathway in respiration is called the citric acid cycle, or commonly called the Krebs cycle. For as long as there is oxygen present, aerobic respiration proceeds and the pyruvate is converted into acetyl-CoA molecules. This acetyl-CoA molecule is the starting point of the Krebs cycle. Each rotation of the citric acid cycle has a net gain of 1 CO2, 1 ATP, 3 NADH molecules, and 1 FADH2 molecule. Almost all living beings rely on cellular respiration. For instance, autotrophs like bacteria and protists are able to make their own food through cellular respiration. Even heterotrophs who can't make their own food, like animals, fungi, and some protists, rely on cellular respiration to process their food. Generally, you can subdivide these into aerobic respiration and anaerobic respiration. As the name suggests, aerobic respiration relies on oxygen. The electron carrier, which is the cytochrome oxidase, is used to differentiate closely related bacteria, like the gram-negative opportunist Pseudomonas aeruginosa, along with Vibrio cholerae, which both use the cytochrome C oxidase, while the gram-negative Enterobacteriaceae, like the E. coli, use different types of cytochrome oxidase. During anaerobic respiration, without oxygen, the inorganic molecules act as the final electron acceptor both in bacteria and archaea. Denitrifiers use nitrate and nitrite as the final electron acceptors, producing nitrogen gas. E. coli, which is usually in aerobically respiring bacteria, switches to using nitrate as final electron acceptor and produces nitrite once oxygen becomes unavailable. Since the primary purpose of cellular respiration is to provide cells with the needed energy, this allows organisms to function. After all, other interventions involving prokaryotic and eukaryotic organisms will not be possible if there is a lack of energy.
ETC, or the electron transport chain, is the final stage of cellular respiration. ETC produces the majority of the ATP with 34 as compared to the 2 in glycolysis and the 2 in the Krebs cycle. It also occurs in the mitochondria of eukaryotes, wherein the inner membrane contains the electron transporters that shuttle electrons to create a concentration gradient. The presence of cristae, or inner folds, increases the surface area that is favorable for ETC. For prokaryotes, it happens in the plasma membrane, while surviving in anaerobic environments mean that they do not need oxygen to complete. Two important concepts to understand are chemiosmosis and oxidative phosphorylation. Chemiosmosis is the generation of ATP from the energy of the proton gradient, in this case, the difference in the concentration of H+ on the mitochondrial matrix and intermembrane space. The gradient is also called the proton motive force, which is comparable to a battery that stores energy. On the other hand, oxidative phosphorylation is the process of using the energy released from the oxidation of molecules to form ATP. It is generally the result of ETC and chemiosmosis. The two crucial molecules in the ETC are NAD and FAD2, produced during the earlier stages of cellular respiration. NAD is excellent in donating electrons due to the high energy present in these electrons compared to FAD2. There are a total of 5 complexes. Complex 1 is composed of flavin mononucleotide and iron sulfur containing proteins. It has NAD dehydrogenase as the enzyme, so it oxidizes the NAD. It can pump 4 hydrogen ions from the mitochondrial matrix into the intermembrane space. Complex 2 oxidizes FAD2, and FAD2 generates fewer ATP molecules compared to NAD. It contains succinate dehydrogenase, which forms a small complex with FAD2. This complex is directly delivered into the ETC by passing complex 1. Complex 3, also called cytochrome oxidoreductase, is composed of cytochrome B, iron sulfur protein, Reas center, and cytochrome C proteins. Complex 4 is composed of cytochrome C, A, and A3 proteins. It contains 2 heme groups and 3 Cu ions. The ATP synthase complex uses a proton gradient to form ATP from ADP and inorganic P. It acts as a generator powered by the H ions that diffuse through it down the concentration gradient. The 2 electron carriers are ubiquinone. Q is a lipid-soluble molecule that delivers electrons from complex 1 and complex 2 to complex 3. Once it is reduced into QH2, it can carry pairs of electrons at a time. Cytochrome C, or Cyt C, delivers electrons from complex 3 into complex 4. It can carry only 1 electron at a time. So in summary, the depositing of electrons from NAD and FAD2 into complex 1 and complex 2 respectively initiates the electron transport chain. Electrons travel from complex 1 or 2 into the Q, complex 3, Cyt C, and then complex 4. As electrons move through each complex, it loses its energy since the energy is used to pump H protons. So as an electron transfers, 1 H+ is pumped out. The final electron acceptor, which is usually O2, accepts the electron. In the case of O2, electrons and protons bind to it to form 2 H2O molecules. The high concentration of H+ enables chemiosmosis for the ATP synthase to generate ATP. The oxidized NAD+ and FAD are recycled for glycolysis and citric acid cycle. Examples of microorganisms. Aerobic cellular respiration is the process of using oxygen as the final electron acceptor. The electron carrier for microorganisms is cytochrome oxidase, and it differs depending on bacterial types. Examples of microorganisms include Pseudomonas aeruginosa, Vibrio cholerae, and Enterobacteriaceae. Anaerobic cellular respiration is the process of using organic and inorganic molecules as the final electron acceptor. Methanogens use carbon dioxide, producing methane, and examples of which are Methanobrevibacter gottschalkii, Methanobrevibacter smithii, and Methanococcus jannachii. Sulfate-reducing bacteria and most archaea use sulfate, producing a hydrogen sulfide, and examples of which are the Sulforomonas and the Sulfovibrionaceae species. The nitrifying bacteria use nitrate and nitrite, producing nitrogen gas. And examples are Theobacillus denitrificans, Micrococcus denitrificans, and Acromobacter sp. Application to society. The majority of ATP is produced due to the ETC. ATP, or adenosine triphosphate, is the energy currency of living organisms. Due to ATP, cells are able to store energy and initiate and complete cellular processes. Fermentation is an anaerobic respiration that uses organic molecules as a final electron acceptor. It is widely used in the food and pharmaceutical industries. This will be discussed further in the video.
The last process in cellular energy production is fermentation. Fermentation is the collective term used to refer to processes that use organic molecules as the final electron acceptor. This process results in the reoxidation of NAD+ from NADH, which will then be utilized to continue glycolysis. Despite this reoxidation, no ATP is directly produced from the process, as it does not involve an electron transport system. Fermenting organisms can only produce a maximum of 2 ATP molecules per glucose during glycolysis. While prokaryotes are prone to using several different methods of fermentation, the two most common are known as lactic acid fermentation and alcohol fermentation. In lactic acid fermentation, one pyruvate molecule and an NADH molecule is converted into lactic acid and NAD+. This is used by animals and some bacteria, like in the fermentation of yogurt and kimchi. During this process, pyruvate is used to oxidize the NADH, producing lactic acid and NAD+. This process is catalyzed by lactic acid dehydrogenase. For alcohol fermentation, we have 2 pyruvate molecules, 2 NADH molecules, and 4 hydrogen ions converted into 2 ethanol molecules, 2 CO2 molecules, and 2 NAD+ molecules. This process results as seen in the production of ethanol. First, pyruvate decarboxylase, an enzyme, will remove a carboxyl group from pyruvate. This releases CO2 gas and produces acetaldehyde. Next, an electron is transferred to acetaldehyde. This produces ethanol and NAD+. The catalyst for this process is known as alcohol dehydrogenase. A number of microorganisms require fermentation to survive, but in consequence also produce a number of different byproducts. These include Saccharomyces cerevisiae, or yeast, which is the common microorganism that undergoes alcoholic fermentation. Lactobacillus, gluconostoc, and streptococcus are called lactic acid bacteria, or LAB. This group of bacteria utilizes lactic acid fermentation and produces yogurt and other soured food products. These bacteria also produce a low pH environment in the body, which inhibits the growth of pathogens. This keeps our gastrointestinal tracts healthy and free from bad bacteria. Another is Clostridium acetobutylacin, utilized in acetone, butanol, and ethanol fermentations, and produces commercial solvents and gasoline alternatives. Klebsiella and Enterobacter utilize butane-diol fermentation and is used in the fermentation of Chardonnay wine. Clostridium butyrrhizum utilizes butyric acid fermentation and is used in the production of butter. Here we have Escherichia and Shigella, utilized for the mixed acid fermentation and are used in the production of vinegar, cosmetics, and other pharmaceuticals. Propionibacterium and Bifidobacterium utilize propionic acid fermentation and are used to ferment Swiss cheese. As you can see from the examples, microbial fermentation methods have been extensively used in the food industry and other commercial products such as pharmaceuticals. Food products such as yogurt and sour food like pickles and sauerkraut are produced from lactic acid fermentation, while ethanol is produced from alcohol fermentation. Furthermore, microbial fermentation has also been used in the identification of microbes in diagnosing certain diseases. Outside of the food industry, microbial fermentation has also been used in the production of biofuel. This has been achieved through ethanol fermentation of products produced by plants. Additionally, microbial fermentation is responsible for the production of chemical solvents such as acetone and butanol through acetone-butanol ethanol fermentation. Certain components of antibiotics, vaccines, and vitamins are products of mixed acid fermentation. Every day, new research and development results are being presented. These discoveries lead to another chapter in the way of human life. Due to the microorganism's electron transfer chain, respiration, and fermentation, there are many changes that happen in human society. Microbiology studies about the processes of these microorganisms trigger and develop new eras of scientific growth that have vital effects in food industry, drug and antibiotics, new treatments, and many. For example, ever since the advent of biotechnology and bioengineering, different microbial strains have been genetically modified in order to be utilized in the synthesis of various important compounds for medical or commercial use. In a study by Nakagawa, a specialized breed of engineered E. coli has proven to be effective in performing total biosynthesis of opiates through stepwise fermentation. These opiates, such as morphine and codeine, are commonly used as painkillers. Usually, they are extracted from plants like opium poppies. Despite this breakthrough, however, yield is still limited and complex mechanisms make it difficult to increase this yield. Therefore, alternatives are needed. This study proves, however, that a good understanding of cellular respiration can lead to many breakthroughs in the field of science. In the end, cellular respiration is an intrinsic and essential part of life. In order for living creatures to function, they need energy. Sometimes this energy may come from consuming external sources, as in the case for heterotrophs, and sometimes it is produced by the cell itself, like with autotrophs. Whatever the case may be, it is respiration that converts this material into energy usable to the cell in the form of ATP. Of the different processes involved in respiration, it is the electron transfer chain, which is the most effective at this, producing the most amount of ATP. However, the ETC also relies on the other pathways to provide it with the necessary molecules for completion. It is the continuous cycling of these multiple pathways that keeps the organism alive and functioning. Through modern science, we have learned to harness these intrinsic processes to our advantage in order to produce items which we can use, such as antibiotics and food products. In the end, this is only possible through an intricate understanding of the many metabolic pathways, how they occur within the organism, and what each component does during the process. And with that, we would like to thank you for watching this video.